Hi, everyone. Welcome. My name is Daniel Weinstock. I'm the Associate Director of Graduate Studies at IACS. Welcome to the IACS seminar series. This is a seminar series where we bring in people from academia and from industry who are working in applied computation, doing really cool things in lots of different areas. Today's speaker is Georg Gerber. He started his academic career at the University of California at Berkeley, doing his bachelor's degree in math and a master's of public health and infectious diseases. Then he came here to Boston. He got his PhD in the joint Harvard-MIT Health Science Technology program and his medical doctorate through Harvard Medical School, where he is now an associate professor of pathology. And he's also the director of the computational unit of the Department of Pathology at Brigham and Women's Hospital. Today he's going to tell us about Bayesian machine learning for understanding microbiome dynamics. Welcome.
Mic operating? Okay. Yeah, I'm going to talk about work going on in my lab on understanding the microbiome, and I'm going to actually go, a bit into the technical aspects, since I'm assuming this is a computational audience. And what I'm going to do that is less typical, I'm going to spend a little bit more time on the microbiome, since I'm going to assume people may not have a lot of background with that. So this is just because I'm a physician, I'm required to disclose companies I consult for work with, but nothing I'm presenting was funded by those companies. Okay, so as I said, what I'm going to talk about is first giving some overview of the microbiome, and then particularly focusing on therapeutics and diagnostics, which is what my lab is interested in, through the lens of looking and understanding the dynamics of the microbiome, and I'll explain to you why that's important. And then I'm going to talk about two algorithms we've developed recently for doing different tasks with understanding the microbiome and ultimately getting towards these therapeutics and diagnostics. Okay, so for those of you who haven't heard about the microbiome, although you've probably heard something about it, but this is the collection of microorganisms that colonize on and within your body. They're basically everywhere throughout your body. And there's some numbers bandied about, but those 1.3x. So you have more microbial cells than human cells in your body. So are you a human or a microbe? Is an interesting question. And also there's about 150 times or more genetic content in the microbes than the human genome. And I'm going to talk about not just counting things, but kind of their function and why they're important. So as I said, they're everywhere. This includes on the skin, where they play important roles, and then through the GI tract, starting in the mouth. The most densely colonized area in your body is within your colon. But they're elsewhere. They're in the vaginal tract for women. They also colonize areas in the airways, etc. And there are different types of microbes in these different areas. That's what the figure is showing there with the translucent body. So sort of what does the microbiome do when you're healthy? One key function is they're involved in metabolic functions, primarily in your gut, but also elsewhere. So they can break down things that we can't. And these are very important sources of energy for animals, not so much humans, but still important. But they also make things that we can't. So there are compounds, chemical transformations they can do that humans can't. A classic example of this is the reason why they give infants a shot of vitamin K is they are not yet colonized with microbes, and so their clotting function can be compromised. You need vitamin K to clot. They're also important, interestingly, in signaling to us. So there's a constant interaction with the host. And that includes things like having motility in your gut, as well as there's really interesting new evidence that they make neurotransmitters that may go to the brain. They also have key roles in protecting against infection, which I'll talk about more, some of the mechanisms of how they do that. And then there's a really interesting role with sort of educating and shaping the immune system. We classically think of the immune system as there to kill off bad bacteria and pathogens, but there's increasing recognition it has this role of actually kind of selecting which beneficial microbes are in your gut. That is not good. Try placing this lower, we'll see if it helps. So in terms of selecting which beneficial microbes are in the gut and preventing sort of overgrowth of things that you don't want. So one of the big interests where I do my work at Harvard Medical School is, of course, disease and how the microbiome impacts this. So there's increasing recognition the microbiome is involved in a lot of diseases. There are some where this is absolutely proven, like dental decay is caused by microbes. Infections, obviously, are caused by microbes. But there's this whole other axis of emerging diseases which are very interesting, like is shown. I can't even see the laser pointer, so I'm just waving my hand. Let's see if I get a mouse pointer. Here we go. So in areas like metabolism, so things like diabetes, obesity, et cetera, there's increasing recognition the microbiome plays a role. Also autoimmune diseases, things like allergies, in fact, there's quite compelling evidence the microbiome is important. So this is becoming a huge area of interest kind of across lots of different diseases. So in this context, there's a lot of work on developing therapeutics and diagnostics, and I'll just go through this a bit for people to understand practically what we're talking about. So if you go to Whole Foods and buy a bottle of these pills, so-called probiotics, what those contain are living microbes, supposedly. Most of them are dead. But it's important to understand those were developed for food. So they're things like they ferment yogurt and cheese, et cetera. They're allowed by the FDA because they're considered already present in food. So they're not bugs that have any particular reason to colonize the human GI tract. In fact, they usually don't, and their benefit isn't well understood. The newer area is the third one down there, which is called bacteriotherapies. And the idea with this is these are microbes that are derived from the human microbiome in general. So these usually have to be cleared by the FDA as a drug. It's a much more complex process, but we think would have higher efficacy. The first one, which I skipped over, though, is you don't have to start with the microbes. The idea you can use what's called a prebiotic, which is something that is some kind of dietary compound. This would be typically some fiber that microbes can break down selectively and we can't. And so you're trying to alter the microbiome for some beneficial purpose. And there's a lot of interest in that. There's also what are called phage therapies. So these are viruses that infect bacteria. There are people looking at those to either treat infections or reshape the microbiome. And then there's kind of just traditional therapeutics, which would be small molecules or drugs. But the idea is this is actually a huge unmined sort of chemical warehouse with the microbes in your gut. Oddly, there are all these efforts, to go to the rainforest and exotic locales to collect new small molecules, but people have not before now really looked so extensively in the gut. So there's a lot of interest in that. And then finally, there's a diagnostics market in terms of can we look at the status of your microbiome, either which microbes are there, perhaps the chemicals they're making, and can we make a better diagnosis or prognosis? So for instance, things like colon cancer, there's quite good evidence you can diagnose that early with microbiome. So as with everything in Boston, there has been a lot of business interest. There's an enormous number of startups in the area. We actually have more, I think, than anywhere in the world that are focused on microbiome. Okay, so kind of out of that setting, along with Lynn Bree, who's a professor at Harvard Medical School in the same department as I am, we founded what we call the Massachusetts Host Microbiome Center. And our objective was really sort of advance the field beyond just these kind of descriptive associations. The microbiome might be involved in something to really understanding the mechanistic effects. And the reason we have the name Massachusetts in there is this was funded by an initiative from the state called the Massachusetts Life Sciences Center. And their goal was really to bring jobs to the state and new industries to the state. So we're open to everybody. We have lots of research collaborations, not just within Massachusetts, but even internationally. And what we focused on here, which I won't go into detail, but is really functional capabilities for looking at the microbiome, not so much sort of commodity things like sequencing and whatnot. One of the capabilities I'll mention a little bit more because we use it extensively in research are what are called gnotobiotic mice. So these are mice that are raised in a so-called germ-free environment. So they're in these little bubbles, and when you hold the mice, you have to go through a glove box. Everything that comes in is sterilized, the air is filtered, etc. The word noto comes from Greek for knowledge, and so what it means is we know precisely what's in these mice, so we can put in a defined collection of microbes and do very detailed studies to try and understand the mechanism. And I'll show you examples of where we use those in studies that, for collecting data for many of our machine learning algorithms. Okay, so let me switch gears to talk about why looking at time for the microbiome is important. This is just a little cartoon of showing the microbiome does in fact change throughout life. I'll talk more about that, but that's not the only situation it changes in. What I really like to start off with, this is just something Albert Einstein I think says better than I can. You can ignore time, but then you're just looking at a whole bunch of things happening at once. It's kind of hard to figure out. So really at the highest level, that is the issue with the microbiome as many other systems. So I wrote a review on this a few years ago. I have a few things updating that as to points, but I think it stands up pretty well. And with the microbiome, this dynamics really all starts at the beginning. So when you're born, you're essentially sterile. There's some debate about some minimal colonization, but there's really not much there. And as soon as you come out into the world, you start getting colonized with microbes. This is not a completely stochastic process, though. It has a lot of interactions with, as I mentioned, your immune system, maturation of your gut, also things like what you eat. This goes on in a, as I said, somewhat stochastic, but with some strong systematic components until about the age of 3, and then your microbiome becomes roughly adult-like. But at that point, your microbiome doesn't stop changing. And there's quite a bit of temporal variability in healthy adults, and I'll talk about why on some of the next slides. But even if you're ticking along perfectly healthy, it's common that something will come along and sort of knock your microbiome off course. And I'll talk about that as well. But that's one of our big interests, is understanding what happens when your microbiome is perturbed, either deliberately or accidentally. There's also been a lot of studies in the last about 5 years where people are developing cohorts of patients who are at risk for a disease, like say diabetes, and they're following people over time. So they're looking at, can they link changes in the microbiome to changes in someone's disease status? And then finally, a particular interest of my lab is we're starting to develop these bacteriotherapies, so again, collections of living bacteria, and can we start to actually predict what will happen when we put those into patients? So what drives these dynamics? The interactions with the microbes themselves are very important. So a lot of what microbes do is compete with each other. They compete for things like food and space, etc. They also secrete substances to kill each other off. There's a lot of bacterial warfare. But they also can benefit each other. So there's a phenomenon called cross-feeding where one microbe will break down a product partially, and then others will continue to break it down in these sorts of change. There's very rich and complicated microbial interactions. The host, though, also plays a big role in this. I already talked about the immune system, but there's also phenomena like hormonal cycles or even diurnal cycles. Time of day, et cetera, has been shown to have effects on microbial growth. Then the environment is a big thing. So when I go and give talks and shake people's hands, I'm exchanging microbes with people. And if you have more intimate contact with people, you're exchanging even more. Our diet also plays a big role. And so this is sort of profound things like someone who's a vegan versus a heavy meat eater, but even more subtle things, like if you eat bread at different times of the year, the flour sources often have different ratios of starch, et cetera, that can affect things. And then finally, there are medical interventions, which I see a lot of. Antibiotics are the sort of the poster child for this, where you're killing off a lot of microbes that are in your gut and elsewhere. But we're increasingly seeing more subtle things like drugs that modulate the immune system, which many people are on now for a variety of diseases, can affect the microbiome. All right, so I'm going to talk a little bit about the experimental tools we use for studying the microbiome and hence the kinds of data we often look at. So a lot of the earlier work, and still very much ongoing, is based on DNA or nucleic acids. And so there's two kind of primary ways we look at this. One of them is called shotgun metagenomics. And so the idea here is you take all of the DNA in a sample, including the microbes and whatever else, you fragment it, and then you just sequence it. A second approach is you look at some specific marker genes, and I'll talk more about this one in particular because we use it a lot. And so you're just looking at a so-called amplicon, or a single gene or set of genes in the microbe. You can also look at microbial gene expression. This is a little bit more challenging than in humans because microbes degrade things very quickly, but it's still possible and very interesting. And all of these have the advantage that we can now throw this on essentially commodity sequencing platforms and produce tons and tons of data. So this is a lot of what you see today in microbiome data. The second thing that people are starting to get into is looking at the metabolites microbes make. And this is really, I sort of view it as you're directly looking often at the products that are the end effectors rather than the genes and everything that are upstream. So this can be very useful. The challenge is the number of chemicals that microbes can make is it's an enormous, enormous number. It's not even known. And the modalities that we use to look at this are things like mass spec, are much, much harder to get this sort of high throughput data that we do out of sequencers. But this is a very interesting and hot area right now. So all of this, though, sort of creates the elephant in the room, which is these big data sets. And I'll talk about how we think about that and what we do with them. Let me just say a little bit more about the so-called 16S, because we use this, you'll see this in a lot of data sets. So this is a gene that encodes part of the ribosome. This is the machinery that makes proteins. And so it's highly conserved in microbes. However, there are regions, if you look closely at the structure there, that are sort of these stems that stick out where there is variability. So the idea is you can find this in every microbe, and you can sequence a portion of it, and it sort of serves as a barcode. And we talk about what are called operational taxonomic units, or OTUs, I'll sometimes refer to. You can think of these as roughly like species, but they're defined actually operationally, or based on the sequence similarity. And so we put together groups of things that are very close, and essentially we call that a taxonomic unit. There are various ways that you can name these and do further analysis on them. Okay, so let me now get into the computational part, which I think will be most interesting for this audience. So kind of what are some of the challenges for dealing with this kind of data? On the one hand, we have a large volume of data, particularly for shotgun metagenomics. This can produce, per sample could be 10 to 50 million reads. But on the other hand, when we look at this in terms of sort of classical time series modeling, where it might be, say, stock market or weather patterns, we have really relatively few time points and often irregular sampling. And why is that? So one of them is, as you can imagine, economic constraints. These technologies are fairly expensive. Some of them are still upwards of $500 or more per sample. But there's another practical reason. So a lot of what I study is in the gut, and so that requires getting a stool sample from a patient. So I've had engineering students in the lab who say, for my algorithms work, I need samples every 5 minutes. And I say, I don't know if I can find you a patient that poops that often. So we're kind of stuck in this world where we have to take what we can get, and you learn things you never wanted to know, but the normal adult, the number of times they poop a day, it varies from like 3 to like every 5 days. And so you kind of have to be able to deal with that type of signal in your data. It's also noisy. This in part has to do with all the experimental technologies I showed you, but it's also a big sampling problem. In that if you're looking at a sample of stool or you're rubbing off an area of the skin, that's a very tiny sample of these trillions of organisms that are there and not necessarily representative. We also have a lot of subject-to-subject variability. So unlike in the human genome, where everybody has essentially the same set of genes, here, which microbes you have in your gut may be very different from someone else's. So for all these reasons, we really want to estimate, when we make predictions, we don't want to just make a prediction. We'd like to estimate how confident we are in that. But given all these factors, that can become quite complicated. The ones in bold here, sort of as a computer scientist, I view they're both challenges but also opportunities. So we are not looking, when we're measuring all the microbes in the gut, for instance, or all the metabolites, those are not independent variables. There's a lot of relationships there, some known, unknown, but we can potentially exploit that structure. The dynamics also, for all the reasons I mentioned, you would imagine could be very complicated. But as I'll show you, that's sort of the art of modeling. With relatively simple models, we can capture a lot of the first, second, or even third order effects. And then finally, interpretability is very important in these models, because ultimately, we really have two purposes. One of them is advancing scientific knowledge, understanding mechanism, and the other is doing things like creating diagnoses. And so just having a black box algorithm is not really what we want in these settings. All right, so I'm going to launch into talking about two of the algorithms we have developed specifically to look at these type of data sets. The first is something called MDSINE, which stands for the Microbial Dynamical Systems Inference Engine, which is quite a mouthful, so we just call it MDSINE. The design part of it is intentional because we really view this as an initial step towards being able to rationally design these bacteriotherapies. So this is a very high-level slide, and I'll take you through more detail on the actual model and algorithm. But just the key is the input to this is some measurement of how the microbiome is changing over time. That can be 16S data, metagenomics data. We also require a measurement of the concentration of the microbes. For people who've worked with next-gen data, you may already be aware of this phenomena, but when you look at next-gen data, it's telling you just the relative amounts of things, not the actual concentration in the system. And this is important if, say, you're giving antibiotics that could be changing 3 orders of magnitude. You're not going to know that from just looking at relative amounts. And then finally, a key thing is we require looking at the system when it's perturbed. Because ultimately what we're trying to do here is infer a dynamical systems model, which I'll talk more about. But if you think about it, in general, the inference task, if you're looking at a system and you're just looking at it at steady state, it's almost like you're just seeing replicates of the same thing over and over again with noise. You really want to see the system. You want to see the transients when the system has been knocked around. So once we have that information, it goes into the inference part of our algorithm, which I'll talk about. And then from that, we can make a whole lot of interesting predictions. And I'll give you some sort of case studies for that. Okay, so when I talk about a dynamical systems model, in general, we're talking about some differential equations model. In this case, we use something called Volterra-Lotka. This came out of the ecology literature. They're so-called predator-prey models. I think it was originally for rabbits and wolves, and you saw these oscillating kind of cycles. This has been extended into the microscopic world. So the equation is relatively simple, although it is nonlinear. And so what we're modeling is the concentration, change in concentration over time for each species in the ecosystem. And we're assuming that has a intrinsic linear growth rate. And then you assume it has potentially all of these sort of mass action or quadratic interaction terms with anything else in the ecosystem. And then we also assume that a perturbation like an antibiotic or something else can alter our growth rate in the system. So why do we want to do this? There's certainly a lot of kind of generic models and statistical models looking at correlation and other things. To my mind, these type of models have some real advantage. So the first is they're interpretable. We get things out like growth rate or these interactions that either have an actual measurable biological meaning or something that we can look at and assign qualitative meaning to. But also importantly, these models can be used for forecasting. Unlike just a statistical or descriptive model, we can ask what-if kinds of questions. So I could say, what if a perturbation is applied to the system that it's never seen before? Or what if we do kind of in silico knockouts? What if I take some organisms out of the system? What would I predict would happen? We can also look at formal dynamical systems properties. And interestingly, these models inherently give us directed interactions or so-called mathematical causality. So these is not as strong as real-world causality, but because we are looking at the aspect of time, and it's one thing preceding another, it's a much stronger statement than correlation. So I think there's a lot of advantages, but there's some real challenges to doing inference. So the traditional method to inferring a dynamical systems model is basically you use numerical integration over and over again to predict what your trajectories are, forecast them, and then you do some kind of nonlinear optimization on the parameters. So this is very slow and not terribly scalable. Kind of in traditional things, people look at systems with 3 or 5 organisms. We're wanting to scale this to many, many organisms. These models also don't do very well in this setting where we may have a lot of noise and poor or limited temporal resolution. And then to my mind, one of the big problems is they're not inherently probabilistic. So a lot of the questions as well as techniques that we can use in a probabilistic system aren't available to us. So this was kind of our contribution to the field in this paper, which was published about 3 years ago, almost to introduce a series of techniques for inferring these dynamical systems that was both more efficient and also putting it in a probabilistic framework. So I won't, in the interest of time, go through this in a lot of detail. For people who are interested, you can look at the paper, but it's essentially a 2-step procedure. The first is we try and denoise the initial signal as much as possible, and we introduce a new technique that uses splines. It's adaptive and also takes care of the count and other statistical structure of the data. Once we get that, then that feeds into a second step in the algorithm, which is to try and infer the parameters in the dynamical system. And because of the structure of these equations, it's kind of cool. It actually is, if you know the trajectory, it's linear in the parameters. So you then have all the tools for probabilistic inference on a linear model. We looked at a bunch of different ways of doing that, and it turns out the one that really performs best is a variable selection type of scheme that's Bayesian. And I'll just pause for a few seconds to say something about that because I think it's interesting. So it turns out that the forecasting problem is actually quite easy because these equations have so much modeling capacity, essentially. So you can learn a system you can forecast even reasonably well. But inferring the actual underlying structure of the system is much more difficult. And that's what the Bayesian method is a lot more helpful with because it gives you, for one thing, clear signal as to which interactions you believe or not that are actually in the network. The second aspect to it that's kind of interesting is that with variable selection, you are structurally removing things from the model versus just with a lasso or other type of regularization. And that also gives you a lot clearer picture of what the actual structure is. So we did a lot in the paper to validate that and sort of show that in the context of this problem. All right, so let me talk about a proof of principle with this model. And this is looking at something called Clostridioides difficile or C. difficile infection. So this is a nasty pathogen. It's the actually most common cause of infection in hospitals today. It causes bad diarrhea that can even be fatal. And it's been known for a long time, actually, almost since the advent of antibiotics, that after people took antibiotics, they were much more susceptible to getting these kinds of diarrheal infections. The pathogen itself wasn't known until about the 1980s, but the phenomena was well known. The other really nasty aspect of this bug is that you can get recurrent disease. And so you treat someone, which is giving them more antibiotics, and they can get this over and over again. There's patients that literally have this for years. And there's about a 25% chance that's going to happen to any given person. So there has been a lot of interest for treating C. diff with something called fecal transplants. It is not literally this, where I hook up 2 patients' colons. I like this drawing, though. It is literally this. So there's brown stuff in a tube, and it comes from this. So you really are taking poop from a healthy person and giving it to the sick person. And nowadays, this is typically done through pills. So this is packed into pills, and people swallow them. Okay, so what do we have to say about this? So it actually is surprisingly effective in certain populations. I'll say a little more about that. But as a physician, this doesn't thrill me. So we really don't know how these work. We don't even know which bugs or even that it is the bacteria in there because we're just putting everything in. There are some significant safety risks. So I used to do transfusion medicine with doing blood transfusions on patients. We had some horrific incidents there with transmitting HIV and hepatitis in the blood supply. I'm very concerned about this because we really don't know what we're putting in people. There also is a grossness factor. That's why I intentionally put that up there. And so you can imagine if someone's had this disease for 2 years, they're probably willing to take this. But the average person, there may be some squeamishness. The other thing that is becoming more evident is these don't actually have a 90% success rate when you start looking at larger populations. People that are immunocompromised have other things going on. It doesn't necessarily work so well. So a kind of a dream in the field is what we would call a precision bacteriotherapy. So this would be some pill with a defined collection of microbes. It might even be specific to the patient. And there's a hypothesis that, so if you have C. diff up there, that's the bad guy with the scythe, and you have a bunch of bugs that are inhibiting C. diff, well, you want to put those back in, but it may also be that you have sort of ones that support the inhibitors. So you want to put in that whole cast of characters to get the most efficacy. All right. So as I mentioned, a lot of what our center is interested in is really drilling down into mechanisms. So we developed a mouse model for C. diff infection that's quite interesting. So we developed what we call the notocomplex, which is a collection of, this was the first version, so it has 13 microbes. These are all human microbes we're putting into mice, but they're human-derived. And we select them in part to sort of span the natural diversity of what is in the gut in terms of both function and genetic capability. So it's an interesting model. What we do is follows. We inoculate mice with this. We let the bacteria grow up and hang out for about a month. And then we challenge with the C. diff pathogen. In mice that have no bacteria in there, they're dead within 48 hours. But in this model, they all survive. They actually do get sick for about 5 days. They get diarrhea, but they all get better. But interestingly, they don't eliminate C. diff. So it's kind of a chronic carriage model. And this is, in fact, what happens in patients typically. Many people don't clear the pathogen, but they're not sick anymore. So we have this model. We collect a bunch of stool samples, in this case 26 samples over time. And then we do the high-throughput sequencing to determine what the population levels are of all the bugs. And we also do this quantitation of the total concentration at each time point. So we kind of take that mess of data and we feed it into the MDSINE algorithm, and lo and behold, this is what we get out, which is like an interaction network. As I said, the edges have directions. The thickness of the edge is a Bayes factor, so that's our confidence that the edge is actually there. Everything shown here has a Bayes factor over 10, so we're pretty confident. C. diff is the green one at the bottom there. And if you look at that really carefully, it turns out there are two we predict suppressive interactions. One of them is up here with a bug called Clostridium scindens. We're really thrilled that that just fell out because this was the one bug where there was known to be an inhibitory effect. But we also found another one up here. And you know, what about this whole web of arrows? When I talked about this kind of network effect, we probably don't want to just put in one bug. We want to put in a bunch of them. So we looked at this and we said, you know, we have this sophisticated coalgorithm. We should be able to do something more interesting than just draw these spaghetti diagrams. So to take you through what we did, I have to just introduce one more concept, which if people haven't thought about this a little while, you may have forgotten. And that's just the idea of what stability means in a dynamical system. So the intuitive idea is simply, if you have an asymptotically stable equilibrium point, if you knock that system, it will eventually come back to that set point. So that's just what's shown here in either a phase diagram or in just the concentrations over time, where you may get some ringing, but it eventually comes back. You can define this mathematically, at least when you linearize the system in a straightforward way. The converse is if you have an unstable equilibrium, if you knock that system, it may never come back. And what that means in practice here is Microbes could either go extinct in the system or they may go to very different starting ratios than they were at. And so none of those things are things we would want in a potential therapeutic. And so we use this kind of as a screening measure to say when we're evaluating therapeutics, we want it inherently to be stable. So this is the kind of analysis we can do. What we ask is the question, let's look at every subset of the 13 microbes we have there and ask two questions. So first of all, is that subset stable by our definition of acetonic stability? And secondarily, what do we predict its ability is to inhibit the pathogen? And so,
how did you get to select these 13 in the first place?
Yeah, so that was a lot of both trial and error, but a lot of it was things that were known about bugs that are important for things like maturing epithelium, important for things like maturing immune system. So it wasn't based on any a priori neurons of C. diff, because that was not known. But it was known about things that seemed to be necessary to constitute a functional gut. And we have systems where we now are up to like 40 and larger ones. But this one, to be quite honest, it worked. We got a little bit lucky. So, to that point, if you look at the actual data, see if I can point here, the actual data up here and then our prediction, if you have all of the bugs present, as I mentioned, you don't clear the pathogen. Interesting, as you start removing them, you have better ability to remove or reduce the burden of the pathogen. So that suggests there may be things in there, for instance, that are inhibiting the inhibitors and that type of behavior. As I said, this kind of network effect. And so we do this we look combinatorially, and we can predict, for instance, down, I really can't see this pointer, I'll try doing it here. Down here, for instance, this is 3 bacteria that are together predicted to be stable and also predicted to inhibit the pathogen pretty well. And so what we've done experimentally is started testing these sets of microbes, and we actually have come up with a sort of a minimal set that will function as a therapeutic. I'm not going to show that data. It's somewhat complicated and also still unpublished. But the short answer is these predictions do give us insight into what will actually work, at least in mice, as a therapeutic. I'm not going to claim a human therapeutic yet. Okay, so this is kind of a cool idea, but it has some limitations. So you remember in the Volterra-Lotka equations, you're inherently looking at interactions with every microbe in the system. So it's in order n squared parameters we need to learn. In a real human gut, you often have hundreds of species present. The other thing, which I talked about a little bit, is we have this 2-step inference procedure where we first denoise and then we do inference on it. And so we're not really propagating error there. As a Bayesian, I don't like that. And then we also aren't taking into account prior biological knowledge, that structure we might have. So all of this led to kind of work on the next generation of this type of model. So this was work that most of what I was showing, this was published at ICML last year. And there were kind of 3 main innovations we made. The first was this idea of what we call interaction modules. So this is a different kind of a dynamical system where we assume that you have variables that act in aggregate. So let me explain what I mean here. So here you see, if I have, say 13 microbes here, you might have this kind of spaghetti network of interactions. What we're hypothesizing is that microbes actually function together as groups. And there's a biological rationale for this. A lot of these interactions are the microbes get together and they produce some substance that acts on other microbes. So in many ways, it's the sum total of that substance that's being made that matters. And so we hypothesize there's this latent interaction structure going on among the microbes that we can learn simultaneously with the dynamical system. And because of the structure, the way we set up this grouping or latent structure, that scales as log n in the number of species in the system. So you're having to learn much fewer interactions in the model. We also fully propagate uncertainty through the model. It has some nice features with that more technical have been helpful. And I think the third one was a big reason that ICML was interested in this. We introduced a new inference technique. So there's kind of an interesting problem here. When you're looking at these dynamical systems models, things should be non-negative to be physically interpretable, but that makes inference hard. So basically we showed a method where you can relax that strict non-negativity constraint and have efficient inference. So I'll just say a little bit about this. This is just how we expanded the model. So we include now making the dynamic stochastic. This is very straightforward. We go from the Volterra-Lotka equations, and we just assume there is a white noise term, and then we simply discretize using a first order type of integration. So the equations end up being very similar to the original Volterra-Lotka, but it turns out this actually makes inference more robust for making things stochastic. So let me spend a bit of time on the actual model just to show you what we did. So this is kind of small, so bear with me, but try and stare at the screen here. So this is basically buried in our model is the Volterra-Lotka equation. So we have the same thing with growth rate, but if you squint your eyes really closely, we've replaced the interactions with each ij with a Cij. So that's the cluster or component that each microbe belongs to. And so if you look at this model with plate model notation here, what we have is this repeated structure over the CI here, over the different components. And the idea is the interactions are between the different components in the model. So how are we actually, what type of prior are we putting on the interaction structure? Well, we assume something called a Dirichlet process. So this is a so-called Bayesian non-parametric model that's over partitions of the data. And so that's how we assume these microbes may clump or aggregate. The other piece that's in here is, as I mentioned, this relaxation piece. So we introduce, here is our latent trajectory for how the microbes are evolving. We introduced this auxiliary variable sort of in between the actual measurement and the latent trajectory. And it turns out this is what enables us to do inference efficiently if we make certain distributional assumptions on it. So for those of you who are interested, again, there's an ICML paper on this with a lot of the gory details that I'll skip for in the interest of time. And so with this, it turns out we can do efficient MC-MC sampling. The idea is you are jointly attempting to figure out which cluster the microbe should belong to and learning all these parameters simultaneously. And then as shown in this figure here, we're also simultaneously learning what the trajectory itself is and our noise model, so to speak. The idea being that you may have outliers in the data, and you would want that to sort of be corrected by what you think the actual dynamics are. So that's what's shown here, this filtering or smoothing. But conversely, if perhaps the model isn't fitting well, you want it to have that as well. And so you want feedback between those two parts of the inference. And in the new model, that's what goes on. All right, so let me take you through quickly a few results here. So the first thing we did is just a very simple synthetic data example. So we said, what if we did have a small system of microbes and there were 3 underlying clusters? Can it actually learn that? And we asked the question, what if we go back to the old model, have it learn that, but don't tell it there's any structure there? Because we want to understand, is learning this structure actually of benefit? So it's really interesting. We see kind of the same phenomena that in terms of being able to forecast, being able to learn that underlying latent structure isn't that important. Both methods can do almost as well. The one with learning the structure does a bit better. But where the model really does better is learning the actual underlying structure. And this is kind of illustrated nicely with this figure. So on this panel up here is what it learns when we tell it to learn the actual structure. And you can see it recovers the actual coefficients as well as the structure quite well. If you don't tell it to learn that, it learns this. And the trouble is, you know, what a lot of people have tried with these kinds of models is they want to post hoc, they put it through one model, then they want to post hoc cluster or look for that structure. And as you can see, you actually don't recover it very well with doing that. whereas by learning them both simultaneously, you recover it quite well. So that's sort of the lesson at a high level with this simple example. So the other thing we obviously did is we applied this to our original data set, and it's interesting. So we end up, instead of that sort of spaghetti network with just 3 interacting modules, C. diff ends up in here, and interestingly, the one that had the strongest actual effect was this one bug I told you about that was already known. But we also see effects with others. So we were very happy with this. This sort of automatically gets us to a known thing and in a very strong way. Okay, so in the last 15 minutes, I'm going to attempt to tell you about another algorithm that is kind of solving a different problem, and if I don't have time to get through it all, I'll just kind of give you the high-level ideas. So the setup here is kind of turning things on its head. This is where we were developing a diagnostic to try and predict who's going to recur with the disease. So here we have a cohort of patients that initially come down with C. diff, and then we follow them for some weeks, and we take stool samples. And the idea is, can we look at something that's going on in their microbiomes to predict who's going to get disease and who won't? So this is kind of a common study design in the literature, where you have some multiple measurements over time for an individual, and then we want to predict perhaps a disease or maybe a classification context, like if people were on 2 different diets or breastfed versus formula-fed babies. So when I first got into this project, I assumed we would use something off the shelf, but my postdoc started looking closely into what people were doing, and it tended to be the following. People would manually pick time windows of interest, so they would say if they were looking at infants, they'd say look at the first 6 months and the second 6 months, and then they would just basically average over that time window and then perform a statistical test for every microbe in the system. And as I told you, there's hundreds there, and it's even worse than that because you can look at them at different levels. You can look at the species level, or you can go up to higher taxonomic levels. So this obviously doesn't really exploit the temporal structure in the data. You can't automatically infer those time periods, and you also can't figure out patterns because you're looking at everything separately or independently. So this led us to develop a new technique, which also has a big long name, but we call it MITRE for short. And its input is very similar. We also are looking at time series of microbiome data, but there's 2 big differences. The first is here, this is a supervised learning framework, so we're trying to predict an outcome or label. And secondarily, we're taking into account prior knowledge. So in this case, the phylogenetic relationships among the bugs. The other big sort of difference between this and traditional models is the type of predictors we learn. So in this case, these are human readable rules that can predict the outcome. So let me take you through the structure of those rules and give you some rationale as to why we did it this way. So the rules are structured as follows. We start with what we call a detector, which has 3 different components. The first is that the detector is looking over some time window. The second is it's looking at a group of microbes, so this can be any subtree in that phylogenetic tree. And then it chooses a threshold. And so the idea is you're saying either if the rate of change of those microbes over a time window or their average value over a time window is above and above such and such threshold, then the detector is true. And then you can chain these detectors together into conjunctions, so you can build up quite complicated nonlinearities, but they still remain interpretable. These also then go into a standard logistic regression setting where you're saying if you have a particular rule, which is a set of these conjunctions is true, and another one is true, each one of those has an influence on the odds of the disease increasing. So it's, once they're in this rule format, it's a standard logistic regression. So this is just a simple example rule where it's saying if the average abundance in this particular time window from 2 to 5 months is above a certain threshold for these particular microbes, then the odds of your disease increase by a factor of 10. You can build these up. This now adds another detector, which looks at a different time window, a different set of bugs. This one's looking at rate of change, et cetera. Okay, so a key problem you could imagine with this model is it could just build really complicated and difficult to interpret rules. So this is where the Bayesian setting again helps us, as we put priors on our concept of what an interpretable rule is. So we want to favor rules that have a short number of detectors. We want a short few rules. We also have priors on the time windows. We don't want things that are like impulses. On the other hand, we probably don't want the entire time interval. We also can put priors on where we want to look phylogenetically. The nice thing about this, though, is if we have enough data, the data can swamp the prior, and so it can tell us, oh, maybe you do need to use quite a few detectors here because it's a complicated relationship, or maybe the time window does need to be short, et cetera. We produced an open source package for this. One of the interesting things, which I'll show you quickly, is we also have a visualization scheme for this, which ends up being important in this setting because it's a fully Bayesian model, so we get distributions of these rules. And one of the big things for people who have worked in the setting of diagnostics or looking for biomarkers, it's often true that multiple sets of biomarkers can give you almost as good a predictor. And there's reasons why you choose one biomarker or another that have nothing to do with predictive capacity, like ease of measuring it, cost, et cetera. And so this exploratory capacity, the idea is you can sort of understand, are there multiple things in your design space that you actually have to play with to make a diagnostic? So this, to me, though, was why this I ended up pushing forward with this project. And so my postdoc went and collected a bunch of data sets. There aren't a ton, but we found all that we could that are time series microbiome data sets, and then compared them against kind of the standard in the field, which has been random forest and L1 logistic regression. People are starting to do deep learning, but honestly, the data is, there's not enough data that really helps much. But anyhow, so random forest had been kind of the top performer, and so I expected them all to look like this, where we did a little bit worse than random forest. And I said, you know, that would be okay, because we're fully interpretable. This was a random forest with like 10,000 trees, okay, and we're looking at, for the point method, the one in orange, that's a single rule. So I thought it would all look like that, and in fact, in some cases, you know, random forest outperformed us. That didn't surprise me. But in a bunch of other cases, we actually do much better than random forest. And that initially surprised me, but if you start thinking about it, you know, random forest is not encoding the temporal information. It's also not encoding the phylogenetic information. Now, we induced features that put some of this in, even for random forest. It still didn't do as well. But I think that actually including more structure, the problem is clear, can actually help in these settings where you do not have massive data sets where it can just learn everything. So anyhow, that was kind of the interesting take-home or lesson there. So this is what our GUI looks like. It's probably a bit hard to see on the screen in a giant room, but I'll take you through it conceptually. So what's shown on the y-axis here is the set of taxa. So this is a phylogenetic tree. And then on the x-axis is time. And so in regions where you have the orange, the saturated color, this is where it's saying there is with high probability, there's a set of detectors as well as a set of microbes that are predictive of the outcome. And so if you click on those regions, it will bring up what the actual rules are. It will also bring up a view on the data as to where the rules are operating. So this one has a detector here, and these are the two different groups and what the data looks like. And then it also displays the microbes. So this one down here in panel E was kind of interesting. So this was a data set where they looked at formula-fed versus breastfed infants. And what the model learned is it said over this initial time window, which is in about the 6-month period, you see in the formula dominant group, you have this group of microbes that is increasing much faster than in the breastfed infants. And if you look at these microbes closely, they all belong to what are called the firmicutes. So there's two major divisions of bacteria in the gut, which is the Bacteroides and firmicutes. And these are in a particular set that are the Clostridiales. And if you look even more deeply at these organisms, these are ones that are more characteristic of the adult gut. So they're good at breaking down more complex compounds, such as you would have in fat and meat, et cetera. So what you kind of see here is what we're detecting is in the formula-fed infants, they're becoming more adult-like, if you will, in the gut much quicker than in the breastfed ones. So you can quickly pick out those kinds of patterns. Also, what's kind of interesting is you see in this particular data set, it really only found kind of one mode in that posterior for good predictors, whereas in this one it found multiple ones that were almost as good. And so that also tells you quite a bit about, as I said, how sort of specific the results are if you're looking for a biomarker, or if you have choices with what to play with. All right, so just to sort of end here with what we're working on now. So I talked a lot about the Volterra-Lotka model. It actually works quite well. I didn't go through some of the validation results, but it's surprisingly good at predicting these perturbations in a very limited setting, though. And we're starting to look at more complex settings, things like looking at, for instance, interactions with phage or virus, and it doesn't work so well. So we're working on more flexible models of dynamics. And one of the interesting things we're pursuing is making the form of those dynamics be non-parametric, so that it's not encoded by a parametric equation. We're actually learning that along with the model. We're also looking at integrating other data modalities. I talked at the start about looking at metabolomics and mass spec, and we think that's very important. We have several studies with strong evidence of that. But it's a interesting and harder problem. So if you look at something like our MITRE algorithm that looks at subtrees, with metabolomic data, you do not have that type of structure on it. It's much more complicated in terms of pathways, and you have many, many more compounds. And so we have to sort of rethink how those kinds of models could work. And then finally, an area that I'm really excited about is we're starting to look at how to model interactions with the host, because that's really what we care about ultimately. And in concert with this, of course, we have to get data to be able to do that. And so I have some projects where we're trying to come up with high throughput platforms where we can start mining that kind of data. So I'll just end with acknowledgments. The work on the MDSINE2 algorithm is Travis Gibson, who has spoken here in the past as well. This is Travis. And then the work on MITRE was Eli Bogart, this is Eli's picture here. And then this is Lynn Bree, who, as I mentioned, is the director of our center, and much of the notobiotic work and all of that sort of thing she's been extremely instrumental in. And then finally, the initial MDSINE work, and I continue to collaborate with him. This is Vani Bucci, who's at UMass, and has just been a phenomenal collaborator. And then finally, lots of funding sources. So I'll close there.
What sample sizes are you dealing with? I mean, in GOBA, I know you have specific studies, but what's the total volume of data you have access to?
Yeah, so it is getting better. Some of the, like the studies I showed here, the largest ones are probably on the order of 100 subjects, and then you have others that are, more like 50. Some of them even going down to 30. People are starting to do studies now where we're getting into the hundreds. But we're still not really pushing the thousands or tens of thousands you have in some of the human genomic type data sets.
So in the Bayesian, when you do the clustering with Dirichlet process, so are you using priors from data in that case, or are you just uninformative priors?
It's pretty uninformative. So
if you use some data to do some naive clustering there, do the clusters come naturally or?
No, they really, so the issue is, and I have another model that does kind of the obvious thing. The obvious thing, let me see if I can get to a picture. The obvious thing you do is to cluster the trajectories. The problem is this is not doing that. That's not necessarily desirable, because if you have different initial conditions, your trajectories may look different, but they still have the same interaction structure. So this is a more general idea than just clustering the trajectories. Yeah.
General question about the probiotic in the market now. There's some food people, the manufacturer added some probiotic. Do you think that's healthy, or do you have any comments about that?
I think it's generally bullshit. So there's been interesting, there's been people who buy the products from the market and then actually culture it or sequence it. So first of all, most everything in them is dead. You have to consider the product is sitting on a shelf for who knows how long, no cold chain. The second thing is they often don't contain what they claim they do. So the concept of purity, there was another colleague I have who did this. There's an analogous market for soil additives. So these things, if you go to like Home Depot, they have things to put in your garden. They claim they have all these beneficial bacteria. He found like none of the bacteria in there. So the trouble is it's a very unregulated market. And the health claims also don't, because it falls into this generally regarded as safe and food, the health claims don't undergo any scrutiny. I can't say to you it cures diabetes, but I can say it makes you feel better, and you know it helps your immune system. And they have all this stuff. If you look at any specific claims, though, they don't have it. So, that said, I mean, I think that some of them probably do have weak effects. And that's what is very suggestive that these, microbes could help. But it's really the matter, we need to look for the right ones rather than just things that are food additives.
Thank you for your talk. I found it very interesting. I'm curious about these microbiomes, if they have, in your opinion, like any impact on the trajectory of species evolutionarily. Like, I think the primary thinking is that genetics is like really what controls things, but is there some Lamarckian thing going on here that we kind of have discredited for too long?
No, I mean, that's a fascinating question. And I mean, there are people that are starting looking at coevolution between the host and the bugs. And, there's pretty strong evidence of that. And so there's people that talk about the sort of notion of the superorganism. You need to actually look at the contained microbes plus the host genome. But there are things, I mean, it's very interesting. You can look at studies where people have seen species that have lost, host species that have lost certain functions, and it's taken over by the microbial species in them. And so you have sort of that level of coevolution going on. Yeah.
Another question on sample economics. Can you talk about the economics of actually processing, say, a fecal sample from a human, collecting sufficient amount of data, doing it, and what does that cost look like, say, with higher volume, or how do you expect it to change in the next few years?
Yeah, so that's also a great question. It is evolving. So I would say right now, the cheapest thing is the 16S analysis, and that in our center for someone who's a affiliate and all the rest is about $50 a sample. And part of the cost is the DNA extraction. It kind of, we don't yet have a lot of ways to make that faster. When you get into the shotgun metagenomics, if you go to I won't mention any names, but if you go to local institutions, that can be more on the order of 500 a sample, but people are looking very aggressively. So they're looking at ways where you can do the actual library preparation much more cost effectively. The sequencing right now does not tend to be the limitation, it's the library preparation. And so, there are people that are driving that down into the, more like 100 per sample. So I do think we're going to be getting into that realm soon where it will be under $100 even to do the shotgun stuff. Yeah.
I think it's known that people in different parts of the world have different susceptibility to disease, and maybe there's a correlation between microbiome and their diet.
Yeah, no, so people have been starting to look at this pretty intensely, and it spans a whole range of diseases. So certainly with autoimmune diseases, there's a great variety of susceptibility. And what's interesting is you'll have sometimes populations, they will move to another country, and their risk starts to look like people in that country. So that suggests there's obviously a very strong environmental component that could be microbiome. The infection story is also the same. And one of the ones that's really interesting is in malnourished children have very high rates of susceptibility, for instance, diarrheal diseases. And part of that impact, even when you treat the malnutrition, so you give them enough food, enough calories, enough vitamins, they may still have lingering increased susceptibility. And so there are people looking at, are there long-term effects, microbiome immune interactions causing that?
All right.
Let's take one more.
Thanks. You mentioned the sources of variability, and one of them is that different people have different volumes and weights of production per day, and different microbial contents in that, and what you get out is X million reads. What is the scale on which you try to measure everything in terms of bacteria per something? And
So, like,
how do you do that?
Yeah, so what is the sensitivity of the assay in a sense, or?
What is, when if you're trying to look at counts or at interactions, it matters whether you're looking at absolute counts or proportions, or so how do you do that in such a way that you're not seeing false interactions because of compositionality and that sort of thing?
Yeah, so in the MDSINE work I showed, yeah, what we do is we measure both the composition and the concentration. And so the units we end up getting are equivalent to colony-forming units per gram of stool. So it's an actual concentration.
Yeah, so I think what you're going to do, we take two measurements actually. So we do the read sequencing, we also do qPCR. We get a quantitative measurement, and then that's how we have a joint model for both measurement modalities coming together to give you an absolute abundance. So that's how we get over the compositionality.
All right. Yeah.